I'm Rachel Fish, and I'm an R&D scientist at ClonTech Laboratories. And today I'll be talking to you about single-cell RNA sequencing with SmartSeq v4 technology. So just a quick agenda. I'm going to start off by talking about transcriptome analysis and why we study single cells. And then I'll do an overview of the SMART technology just to make sure we're all on the same page. And then I'll talk about the single-cell capable mRNA-seq using the SMART technology, using the SmartSeq v4 ultra-low RNA kit for sequencing. So we can do transcriptome analysis with next-generation sequencing, and that's a technique that's referred to as RNA-seq, which can produce millions of sequences across complex RNA samples. So we can do many different things with that. We can look at gene expression, discover and annotate complete transcripts, discover and characterize alternative splicing, a number of things. And in recent years, more and more people have been interested in investigating these applications at the single cell level. So I'm not going to go into a lot of applications, but I do refer you to a very nice review that just came out recently that discusses the technology of single-cell RNA sequencing and a number of the biological questions that people ask. So why do we study single cells? Well, we know that during the cell cycle and during different processes, gene expression can vary. But what about for other genes? We do know that gene expression can vary significantly between cells, and that the average expression of a population doesn't necessarily correlate with gene expression at the level of an individual cell. So what we're showing here is a histogram of cells, single cells, and we're looking at the expression of actin, what is usually considered a housekeeping gene, and this is on a log base 10 scale. So we can see that even the expression of actin varies by about a 4-fold order of magnitude for single cells. Another thing we might want to do is cell identification. So here we're looking at a study by Ramsgold et al., and this is one of the first studies using single-cell RNA-seq, and this actually uses our first-generation SMART technology. And in this case, they were able to categorize individual cells in a population according to their cell line of origin based on the transcriptome. So they had 12 individual cancer cells, and they looked at the transcriptome, and by a type of PCA analysis, they were able to see a separation of the single cells into their cell line of origin. One question we ask is, does the variation we see in single cells, does it recapitulate what we see in the bulk sample? So what we're looking at here is a study from Steve Quake's lab using the Fluidigm C1 system, single cells, and what we're looking at on the x-axis is an ensemble of the gene expression of those single cells, and on the y-axis of bulk. And what you can see is that there's a pretty good correlation, which really says that the variation that we see in single cells is present in the bulk population. So I've mentioned SMART technology a couple of times, and so now I want to talk to you about some of the key aspects of this technology. So we can start with total RNA or actual whole cells. And what happens is that we use a SmartScribe reverse transcriptase to make the cDNA. And what happens is when the transcriptase reaches the 5' end, it adds a few non-templated nucleotides at the end. And by virtue of the addition of these non-templated nucleotides, a specially designed oligonucleotide can specifically hybridize to there, and this basically forms an extended template. And the reverse transcriptase can then actually switch the template and continue transcribing and replicating it to the end of the 5' end of the RNA. And it is this mechanism that gives SMART its name, which stands for the switching mechanism at the 5' end of the RNA template. And then since we are able to define both the priming sequence and the switching oligonucleotide, we can add our adapter sequences of interest. And in this case, we can add special sequences that enable us to do PCR. So as I said, so since we're getting to the 5' end of the template, we're generating full-length cDNA. And what you're looking at here is a bioanalyzer trace of the cDNA produced. And this really reproduces what the full-length RNA profile looks like as well. and then we're amplifying. So how does this fit into your workflow? So in green, you can see the workflow that I just described, the cDNA synthesis and the PCR. And like I said, we can take total RNA of good quality, this is RINs greater than 8, or whole cells. You can go into this workflow, and then because we're generating full-length cDNA, for most platforms, you do need to fragment that cDNA to appropriate sizes and add appropriate sequencing adapters. So we have protocols that allow you to either shear and go into a library preparation kit for ion torrent, or you can go into Illumina workflows by either Covara shearing and then using a low input library preparation kit to add the adapters, or you can use the Nextera protocol, which does tagmentation, which does the fragmentation and the adapter ligation in one step. And then, of course, you can go ahead and sequence. So what I've described so far is simply our SMART technology, but recently we came out with the SMART-Seq V4 ultra-low input RNA kit. So this is our latest generation. And what we did here was we further optimized the template switching oligo. We've incorporated LNA technology that was developed in Ricard Sandberg's lab at the Karolinska. And then we also use our own proprietary knowledge to further develop the template switching mechanism for further improvements. So in this way, we were able to improve the sensitivity and the reproducibility. We measure sensitivity in terms of the number of genes identified. And with the increased sensitivity, we're actually improving the reproducibility of especially the low expressors. And then we're also able to get a higher yield for a given number of PCR cycles. So this gives you a little bit more cushion if you have single cells with varying RNA content. For the same number of PCR cycles, you get good yield. And then we've also further simplified the protocol. So let me go quickly over some sequencing data. So what we're looking at here is 3 different cDNA synthesis methods: the ultra-low V3, which was our prior generation kit, the SMART-Seq 2, which used the LNA technology, and then our SMART-Seq V4. And since our goal is to really remove the technical variability, or to reduce the technical variability as much as possible, rather than using cells during development, we use a control RNA and we use it at inputs that are approximately equivalent to the amounts found in a single cell. So in this case, I'm showing data from 10 picograms of mouse brain RNA. And really what I'd like to call your attention to here is the number of transcripts identified. And you can see that with the SMART-Seq V4 technology, we're getting a significant improvement in the number of transcripts identified over the other 2 technologies. These other statistics I'll discuss a little bit later in the context of cells. So for those of you who aren't familiar, these are correlation plots. And we're looking at here, these are all from the data that I showed on the prior slide. So this is 10 picograms of RNA. And we're looking at is 1 replicate on the x-axis, and we're looking at the expression level in terms of FPKM. And on the y-axis, we're looking at the second replicate. And what you can see on the top is we're looking at both a Pearson correlation, which is R, and a Spearman correlation, which is rho. And you can see that with the SMART-Seq V4, we're getting a very nice both Pearson and Spearman correlation. But for those of you familiar with these plots, we do tend to see that the high expressors really tend to drive the correlations. So what we did is we removed those high expressors, that is anything with an FPKM greater than 100, and then we recalculated the Spearman and the Pearson correlations. And that's what you see down here. So now you can see we have a very nice Pearson correlation of about 0.74 and a Spearman correlation of 0.6, which is significantly higher than the other 2 technologies. And the other way just to see this qualitatively is kind of looking at the spread down at the bottom here. And you can see that we have tightened up this spread for low expressors. And what we really think we've done is we've really improved your confidence by about an order of magnitude in terms of the FPKM. So now that we feel like we've really reduced the technical variability to quite a significant amount, we moved on to real samples. So what we're looking at here is single cells. Here we're looking at HeLa cells. And we're just looking at bioanalyzer traces from single cells. We have noticed that we do get a variable cDNA yield, and we did notice when we did the experiment that cells did vary in size, and we think there may be a correlation there, but we really haven't pursued this further. And then what you're seeing on the right is a bulk sample with either 100 or 1,000 cells. So in the next couple of slides, I'll show you data from these preparations. So again, here's sequencing data from, again, this was from intact HeLa cells going directly into the protocol. And here are the different replicates that I showed you on the prior slide. And first you can see that we do have a number of transcripts identified, quite a good number. It of course does vary, and we do see that with higher yields, samples 4 and 5 were the samples that you saw with the higher yield, we do get higher numbers of genes identified. But I'd like you to see we have very low mapping to ribosomal RNA and also very low mapping to intergenic regions. So now if we next look at correlations, here we're looking at single cell correlations. So we're looking at each of the single cells against one of our other single cells. And you don't expect a perfect correlation because of course there is variation in single cells. But in spite of that, we're actually still seeing very nice correlations in both the Pearson and the Spearman correlation. So what we're seeing here is both the technical and the biological correlation. And then we also compared each of those single cell data to the bulk samples. This is 1,000 cells. And again, we wouldn't necessarily expect an exact correlation because it is only a single cell, but we actually are getting very nice correlations. And of course, when we compare a bulk sample against the bulk sample, we're getting a very tight correlation. So we can also look at gene body coverage, and again, these are the same cells that I've been showing you. Here we're looking at gene body coverage across a normalized transcript length, so we've normalized all the transcript lengths to 100%. So we're looking at the 5' to the 3' end, and then normalized read coverage. And we have very even and reproducible gene coverage, and this also recapitulates the bulk sample. So we're really not losing any information across the transcript. So I really didn't have a lot of time to talk to you today, so I've really only talked to you about one of our kits. But if you're interested in more kits and solutions that ClonTech has for next generation sequencing, I refer you to our next generation sequencing resource portal, where you can see technical notes, webinars, posters, and the like. And if you want any more information, you can go. We are at booth 16. If you want any additional information. So that's all. Thank you very much.
I would love to know that what's the bioinformatics tools that you use exactly for mapping, if you have any preference, because single cell is, to me, I think has to be really treated different than bulk RNA-seq analysis tools that people use generally. You mentioned FPKM, but I would love to know for V4, beside proving the kits, if you can come up with some, can give me some suggestion which tools I should use that can help me for the data that actually looks much better. I can get the better final data also.
Well, thank you for the question. So I'll say upfront, I am not a bioinformaticist. So actually, we recently did get a little more bioinformatics help, but most of these data that you've seen were actually generated with, it's actually a platform, a different software. It's called CLC Bio, but we're actually starting to use other platforms and just starting to use like TopHat and other freeware so that it's more similar to probably what our customers would use. So we are moving towards that. So we actually don't use any special tools right now, but we're trying to learn more about what customers are doing and what kind of tools we might be able to use and to show data from that. So, but this is really just very simple analysis.